Good morning, everybody. This is Dr. Malini Chatterjee from OneMed from New York. OneMed is the home of OneMed Research that covers small healthcare companies, and OneMedRadio, it's the home of OneMedRadio. Today we are very fortunate to have with us Dr. Craig Dees. Dr. Craig Dees is the CEO of Provectus Pharmaceuticals, a Tennessee-based company. Provectus Pharmaceuticals is ticker PVCT on the bulletin board with a market cap of $105 million, average trading volume roughly 200,000 shares a day. Provectus Pharmaceuticals, their story intrigued us in November, December of last year when we initiated coverage on the company in December 2011, and this is a company that uses a compound called Rosebangle, which has actually been approved by the FDA many years back as a liver diagnostic agent. Provectus has found a new use for Rosebangle in the oncology indications and in dermatology indications. Today, we are fortunate to talk with their CEO about the oncology indication, particularly metastatic melanoma, as Rosebangle has recently received the blessing from the Rosebangle and Provectus have recently received blessings from the FDA for the final design of their phase 3 clinical trial. They are in the process of drafting the documents that will be sent to the FDA for special protocol assessment, and thereafter, Provectus's phase 3 trial in metastatic melanoma can begin. This is an important milestone for the company as it takes the company from phase 2 to phase 3 levels. It is proof of the pudding, and it is the time when larger commercial partners start to get interested in the story. So there is much to learn from this story, and without much more talking on my end, let me turn the mic over to Dr. Craig Dees. Dr. Dees, good morning to you.
Oh, good morning to you.
Dr. Dees, I hope I did somewhat of a fair and somewhat of a good justice to the description of your company.
Oh, you did very well indeed. As they're starting to actually mature quite a bit now, it's hard to describe us as a startup as we're going into the final phases of our first indication and have others coming online.
Dr. Dees, before we go into the main topic of today, that is the press release that you put out on January 18th about the FDA coming to a consensus with you about the trial design for the phase 3 trial of Rosebangle in metastatic melanoma. Before we go into that part, let me just ask you a little bit about metastatic melanoma itself, the incidence, the prevalence in the U.S., and the incidence by stage. I know that your trial, your phase 2 trials have targeted a certain stage of patients. Now, if you were to stick in the phase 3 program with those same stages, what is the backlog of patients that you will have available to be treated by Rosebangle at the time of its launch? Also, another thing that intrigued me from one of your slides is that while colorectal cancer and all other forms of cancer are somewhat reducing in their incidence over the years, metastatic melanoma continues to rise, and this is National Clinical Institute data from one of your slides, and I'm curious why that is.
Well, first of all, let's explain what we're looking at here. It's not just have to look at a bigger picture than just melanoma. That we have these hallucinogenic xanthines, and not just one. We picked Rosebangle first. Have what's probably the closest thing to a vaccine and a broad-spectrum antibiotic all rolled into one in a small molecule drug, and it just doesn't treat melanoma. Now, we picked melanoma for a wide variety of reasons as the primary indication, but there are other trials going on too. As you mentioned, the one data that's from the slide in liver cancer, and we're working on design for pancreatic cancer too, because every indication we have is that we haven't found a cancer it doesn't treat yet. I just looked at the treatment, the slides on a horse with a huge squamous epithelial tumor, and they're treating other animals, and they've just treated some other patients with squamous cell, several patients down in Australia. That list gets broader and broader. So the picking of melanoma had quite a variety of reasons in it. One, because of particular regional problems with it in Australia. It gave us a small segmented market there that we addressed very quickly if we needed to. Again, as you mentioned, the incidence of melanoma is growing too. In the vast majority of other cancers, prostate and others, the decrease in mortality ranges from a few percent up to 50% in cervical cancer. And that particular slide you mentioned came from a melanoma researcher at Johns Hopkins. He says, I'm getting very discouraged because you look at melanoma, it's up 25 or 30%. And so both the incidence of melanoma is going up and the mortality is actually still increasing with the things you have out there. Now, the reasons for that are probably many. One, for example, the European market seems to be going up tremendously. And from what people tell me is because they're going in vacation areas where there is sun, down to Tenerife, other places, into Thailand, and getting exposed to sun, which they weren't much for. If that's true, I'm not sure, but that's what I've been told by investigators in the field. And so that's better looking at the numbers. There were 62,000 cases in 2008, with 8,000 deaths in the U.S. and about 50,000 dead across worldwide in there. But looking at the trend is a much better indicator, and the trend is than just trying to pick specific numbers. And the trend is up tremendously in both mortality and morbidity. And then you also look at other factors, say as what the competition is in there, the very underserved market, the things that are out there do not work well, if very much at all, unless you catch the disease very, very early and you can remove it surgically. So, again, what the trend is as a market indicator, the regulatory paths in certain areas, we chose that as the primary indication.
Dr. Dees, so your phase 2 studies are based in patients with stage 3 and early stage 4 disease. What I wanted to ask you is, with greater awareness of melanoma, what is the incidence of disease by stage? And why I ask you this is because if your labeled indication in the future continues to be stage 3, stage 4, or stage 3, early stage 4, what is the accessible patient population? If the incidence is 62,000, what is the accessible patient population to you for those two stages?
I understand your question very much, but I actually look at a much bigger picture than what we've chose to address for licensing strategy. Our initial claim was to get a very narrow window to get it approved quickly in there. But I believe that the patient population is going to be much, much bigger in the end, not just as those label indications in there, because what the drug does is that it seems to appear in quite a number of patients generate a very, very specific immune response. And this was just shown in a recent science paper in mice verifying how autophagy drugs, what they do, generate this immune response. And therefore, as a number of our physicians said, that even if you're contemplating surgery on your patient, that you should put this drug in first. And so I look at it that the accessible patient population is way, way bigger than the label indication eventually, that the side effects of the drugs are so minimal in there. And again, it's not priced yet, but price is becoming very, very important in a number of the recent drugs. And they're horrifically expensive, and they're prohibitively so that people aren't using them. I can guarantee you, although this is not priced yet, that that's not going to be a problem here, similar to the other drugs, that we can underprice it and fit the markets. And still, so I think that the accessible patient population is way, way broader than the label indication.
Now, you had mentioned in a few minutes back that despite of there being approved drugs in this indication, the market continues to be underserved. And we do know that dacarbazine, DPIC, and temozolomide, TMZ, have been used to treat patients with advanced melanoma for a long time. Recently, Bristol Myers got Yervoy approved, and Roche got another drug approved, Zelboraf. How are those two agents doing in the market now? What kind of pockets of patients are they being used to treat? And so with a total of 4 approved agents, what seems to be the problem? Why is there such an unmet medical need still?
Well, first of all, those that were classically treated on very late-stage patients in their trials, and they're rightly so. And again, there's a factor coming on in oncology that before hasn't been in the past, and that's cost. And that to treat something where a few $1,000s worth of surgery in early stage and to come up with a $100,000 plus treatment drug, that there's a problem in trying to use those and to move them into other stages. So they will classically be confined to very late stage disease where you really don't have anything else. And even then, if you compare to the initial study of ours of side effect, now a trial's not good till you run them side by side where you get accurate data, same patients, population, same investigator, and that's going to be very hard to even do with us because ours is a very different delivery route than theirs. The side effects of ours appear to be minimal compared to everything else, so the safety profile feels to be way better than things that are already out there in there. And again, not priced yet, but we're not going to have that prohibitively expensive cost attached to us. We're very frugal in running the trials. We're very good at it, and how we're proceeding to market, and we won't have to amortize billions and billions of dollars across the drug price. And also out there, one of the current trends out there is a very precious sort of, it's designer drug sort of thing genetically for the patients in there, and that is the hot buzzword trend that people say these days. But again, when you look at the number of patients, when you have a specific genetic mutation in there, that is a small fraction of the patients in there that it addresses too, so that doesn't affect us at all. And second of all, people forget that those drugs, if you look at the trials, they relapse almost immediately, and the relapse rate is virtually right close to 100% after just a few months. And I've heard one of our physicians say to me that the disease is 10 times worse. And so those, even the new drugs, being both cost, side effects, and the patient population is addressed, I don't believe will significantly impact us in any way.
Got it. And just to have some numbers, how are Yervoy and Zelboraf priced? Like, what exactly are their prices?
I haven't looked exactly at their pricing. I just know that they're up very, very expensive would be the best way to put that they're highly expensive. In fact, I don't even think they've been approved, some of them in Europe and priced yet. And Europe appears to be the growing melanoma market in there.
Yep.
And so we will move into there for trials too, but they're very expensive.
All right. Now turning to your drug, Rose Bengal PV10, which is the main topic of our discussion today. Can you spend just a couple of minutes talking about the mechanism of action? That is, why is it it is a chemical moiety, so it is a chemotherapy, but given its route of delivery and given its mechanism of action, how this is really like a targeted therapy. Can you talk about that a little bit?
Yes, I can. In fact, it is very, very different. It's paradigm changing. First of all, it's going into the tumor, which is one way it helps it via route of injection. We're not systemically bathing organs. I don't care how good your drug is. If you don't need to bathe the kidney with it, don't do it. You're much better off. But this drug is very, very specific in its targeting compared to virtually anything else out there. It's almost absolute for diseased tissue and doesn't touch normal cells right next to it. So that allows your safety profile to go shooting up, but it also pushes up your efficacy profile because it only hits what it's supposed to. And that's very, very unique, and that's been the underlying 900-pound gorilla in cancer therapy for so many years, is the lack of specificity of our agents we use. Now, what it does also, it kills in a very different way. In fact, you might say it doesn't do the killing at all. It kills by autophagy, which we've showed years ago, that what it does is it partitions, which is the right word, moves into the disease cells only, And it has affinity for a small organelle called lysosomes. And these are full of acid hydrolases, degradative enzymes that cut the macromolecules down to usable components in there. And so it either causes them to leak or explode, and those molecules are out. They start to Faji to digest, to literally hydrolyze the components of the cell. It's a type of self-destruct mechanism in there, a very particular type. And that very particular type of self-destruct in there generates, as was just recently published in Science magazine, a very, very specific type of anti-cancer system-wide immunity. And we've known this for some long time. And so not only does the cell that's induced to kill itself die, but in many cases it goes out, the body, this immunity is generated, and it goes finds the tumors elsewhere at very remote sites. I showed slides the other day of brain metastases disappearing, lung metastases disappearing at remote from the drug action. And again, this is due to a very, very specific type of anti-tumor immunity when a cell dies this way. So it's not a poison. The vast majority of our previous drugs are poisons or toxic agents. In fact, a good chunk of them are the absolute worst carcinogens on the planet themselves that go in and destroy DNA or mutate it in some way or interfere with it chemically in the processes, and therefore are carcinogens in their own right. This drug really doesn't even enter the nucleus. It tells the cell to kill itself, induces it to literally self-dissolve itself, self-eat itself. So how the cell dies is very, very different. And so that's a tremendous factor in why you get the results you do with this class of molecules, is that you're inducing a self-destruct mechanism, and one that generates a very specific remote anti-tumor immunity.
Got it. And since we are here discussing Wednesday's press release, before we go into the nitty-gritties of or whatever level of discussion we can have within your disclosure parameters about the phase 3 design, can you spend a couple of minutes talking to our listeners, to your target investors, about the results that you saw in the phase 2 study of rose bengal and metastatic melanoma, the response rates, the CRs, the PRs, the progression-free survival versus historic numbers, the bystander effect, the effect in nodal and cutaneous disease. Can you just sum it all up for us in just a few minutes so that we have a historic look back?
Yes, we're using objective response rate, which is a combination of what you designate as a complete responder and a partial responder in there. And in our phase 1 trials, that we were around 60%, that objective response rate. Now, it's fraught with peril to compare against other responses in the past unless you're lining up side by side, same patient group, same route of delivery, same investigator, because biological variability is so great in there. And so you can do it very roughly, and so I'll do it roughly that way. And the objective response rates with historical other things are very low and compared against this in the past. But again, side by side would be required to compare them directly. Now, when we moved to phase 2, that in the first 40 patients, they're so almost identical, the objective response rate at about 60%. We almost couldn't believe it, that it was so in there. We looked at each other and said, we're going to have to do something ethically here that will take we're going to change the trial parameters. We're going to start taking much later stage patients in there. And that lowered the overall objective response rate some when we went to much later stage patients and included them for the last 40. But ethically, we figured we had to do that. Plus, we needed to know what the breadth was going to be. But again, if you segment out the same similar patient population, the stage 3, early stage 4s, the reproducibility, repeatability of this, when their stage and grade are very similar and their runs almost identical in there. In fact, it's so identical, you look at statistically as a scientist, you look and say, it's hard, it is so consistent. That's almost unbelievable to yourself, but it is very, very, very consistent on what it does, the reproducibility.
Now, let me ask you, your progression-free survival on the phase 2 study was 8.2 months. How does this compare with if you were able to magically do an apples-to-apples comparison, how does this compare to what progression-free survival is seen in a comparable patient population on the current agents, DTIC?
Well, if we look at that, again, like I say, it has to be a very blue sky guess because they aren't running side by side, again, in the same hands. And again, ours goes an inch or two more. We have a different route in there. But we believe that the comparators will fail in just a couple of months, very likely. It'll be a very, very short interval compared against what we already see.
Got it. And also, in an earlier part of this conversation, you had indicated that to your early steps into the market and your early labeled indications may be for a smaller band of patients, but this drug can actually treat a much wider patient population. Now, when you start thinking about phase 3 design, will you do the phase 3 trial in nodal and cutaneous disease, or would you open it out to the entire patient pool, even with distant metastases, knowing full well that in the phase 2 study, the nodal and cutaneous disease patients had a far greater, had a somewhat greater progression-free survival number?
Well, trial design, you do one thing there, and many times what you do for the patient, that in the trial design is very likely to be limited in the trial to certain stage 3 patients or early stage 4 patients, very likely to be limited there. But also, we have always, at request of both patients and physicians, which I very reluctantly did, was made it available by compassionate use for any other patient that might not fit the clinical trial designs, and that was due to the request from the physicians that while we were not running trials for other patients, that they wanted the drug available to them, the patients wanted it. So it still remains available for compassionate use in ones that might not fit the trial design in both a large number of sites in Australia, MD Anderson, St. Luke's, Louisville, Kentucky, and so we have made that available and will continue to do so for any patients who fit the trial parameters. Got it.
And on your January 18th press release, you talk about reaching a consensus with the FDA on the phase 3 trial design, and then later there is a mention of another phase 3 trial, a separate one for Australia. Now, how are these two trial populations same or different? For the U.S. study, what kind of patient numbers do you envision using? And, given that Yervoy or Zelboraf cannot be used because it's only for a targeted patient population, but given that Yervoy is in its early days in the market, what is a fair comparator to use in your phase 3 study?
The comparator, and that is a very, very hard technical question as for medical scientists, because this is a paradigm-changing thing. How its route, how it goes in is very, very different. It's not systemically applied drug in there, correct, which actually helps it and generate the immune response. It helps it in its safety profile. And so it's very likely that the standard of care drugs, either temozole or DTIC, are likely to be the comparator arm in there just because they've been used so heavily as standard of care approved drugs. There's been other suggestions out there that perhaps we should buy physicians, which amuses me, some that don't run trials, that perhaps there ought to be some interlesional GM-CSF, and you have to point out to them those are not approved therapies, not a good comparator arm. And so, but that's what it's likely to be. As for patient numbers, that's another interesting thing on how much numbers, because it will be statistical endpoint when it stops, of course, against the comparator arm. And for myself, and speaking for myself, that I hope it stops, the committee stops it very, very early. That I think that when they look at both the safety profile and against the comparator arm, that right now the estimates are roughly around 300 patients, but I'm hoping that it stops in a very short period of time. That from historical performance, again, a very fraught with peril extrapolation, but it is so different from the others that I'm hoping at the early look backs that they are statistically so different, performances so different from safety and efficacy that it's stopped early.
Got it. So your trial
you can't guarantee those things, but that's what I'm hoping for.
Okay. But so as I was asking, so would your trial be set up to have an early look at the data for like an interim analysis? Because if
there will be interim analysis for safety and efficacy.
Got it. And now, you bring it up, and this is a very important issue. DPIC and temozolomide are IV agents, and yours is an intratumoral agent. So would this have to be an open-label study?
It's almost impossible to blind a study with this. The
Correct.
drug is very bright red. The physician will be able to see it. The patient will be able to see in there that he's getting it by different routes. So blinding it is not really a feasible thing to do, and also because of route of delivery, how the delivery to the patient is. And so those factors have been in there heavily in consideration going back and forth how we were going to do the designs, that it is very, very different than what's out there. And being a pioneer sometimes makes it a little harder. If it was something that went systemic, it wasn't colored, and had all these side effects, the design would actually be easier.
Right. The other question that comes to mind is the company has spoken a few times about an endpoint that might be a modified progression-free survival. How is a modified progression-free survival different from just your everyday progression-free survival that oncology trials use?
Well, again, it's how you define progression, and that's been one of the issues going back and forth with us asking guidance of the Food and Drug Administration and with our physicians groups, our clinical trial monitors, and a lot of people involved in there, because, again, you have an intratumoral agent going in, and what your claim is, what it's doing, and what progression is becomes a very interesting technical issue. So we've gone back and forth on that. And I think that we know what we're going to propose, but as we said in the press release, this is an iterative process. We'll be filing the documents in there, and it may change a little bit yet.
Okay. So you're saying that because the agent is intratumoral, what is progression is defined differently from if the agent were IV. Is that the way to look at it?
Yeah, like I said, there are a lot of parameters of this is so different in both its performance, its safety, efficacy, how it's delivered in there, that it makes the design quite a bit more, have to have a lot more discussion with both regulatory authorities and actually those that have to run the test. They have a great input.
And this is why we're seeking a special protocol assessment. Because being a pioneer is not easy. The other question about the trial that I have for you is, would there be any need to, after the primary endpoint, would there be any need to have a co-primary endpoint for survival such that the trial continues, although you file your data for accelerated approval and what have you, the trial needs to continue to run for a survival endpoint?
Well, eventually, with any drug, even though these surrogate endpoints we all use to get them to, people forget many times it's very different than an indication where, that it might be itching you in the derm thing or your skin is bleeding. These are patients that are sick and have a lethal disease. And so you have to watch in these and do very different. And ultimately, you will watch whether now or later, you will watch for overall survival. Got it.
But you don't think it will need to be a co-primary endpoint?
No, I think it will. Initially, I'll have to go back and look and see what is actually proposed in there, as it's been going back and forth. I'm not sure. I'd have to ask Eric whether that's built into this or whether we're going to do it later. I'm not sure at this moment. But eventually, you will always look at overall survival eventually in any cancer trial.
Of course. And the other thing is that given historic studies in melanoma, more recently for the recently approved agents, your own phase 2 study, how much of a time do you think this trial could take from the first site opening up to the last patient, last visit, and release of data? The end, if your drug is as striking as it was in phase 2, then there is every reason to believe that the business part of the trial is slow. Is quick to turn around. But what do you, when you do your analysis from opening centers to release of results, what is a time frame that you can think of?
It's very, very hard to predict those in there because patient acquisition, that these are not laboratory animals. The physician has a choice whether he puts the patient in a trial. The patient has a choice whether they come into the trial. And there we're talking human beings here and their welfare. And so even those factors on a major of choice. So it's not just the technical aspects in there. You can't order everybody into the trial like you can a laboratory animal. So it's hard to predict in there, but again, with me and there, that I think I have my hopes that it's going to terminate very, very early, but many of this will have to watch patient acquisition. We'll open more sites than we have done previously to try and go with all possible speed.
Okay. And my last two questions go away from the clinical trial, and now they go into the dossier that you will eventually file for approval. And not many people know this, but Rose Bengal is an approved agent. And when we look at Provectus, it has the fact that Rose Bengal is an approved agent, and many of its safety-related studies and preclinical work and the animal toxicity studies have already been done. Those things have to be featured prominently, as they would reduce the time and the labor involved in filing the final FDA dossier for approval of Rose Bengal. So, Dr. Dees, can you just talk about what parts can you skip in the final dossier because those things are already historically on file from the historic approval of Rose Bengal? What steps will you not have to do?
We very, very specifically chose Rose Bengal. It's not our only halogenated xanthine. We have quite a suite of them. 12A, B, there's quite a suite of them. But we picked this one very, very specifically because of its long use in humans as a diagnostic agent. And better yet, with the radiolabel agent, there was a very big, to use the parlance, unlabeled cold loading dose went into these patients. And so that helped us tremendously at where we could start going into our trials, at what levels we could start putting the drug in. To my knowledge, there hasn't been even the faintest complaints. Me, who's worked in the over-the-counter cosmetics industry, I've seen many more responses back for eyeliner and problems than I have ever seen with this agent going in. Its safety is very, very good. And even then, as we're going forward in filing, so that is just in the overall climate, we very specifically chose this for that reason. Now, what people don't know, that we've been simultaneously running a number of tests for the Food and Drug Administration in these filings. For example, using more modern techniques, we've run a Comet micronucleus assay has come back. We've run a reproductive tox assay that is complete. We've run a new modern Ames test that's complete. And the drug continues to perform in these tests very, very similarly to the human trials, that its safety is just unparalleled.
Got it. Okay. My last question to you is that upon receiving the special protocol assessment by the FDA, if all goes well, and you will get that, your company has always indicated the need for a commercial partner in the future. But would you, in the interest of time, would you start rolling out clinical trial work, the CROs, the enrollment of the trial itself, with or without a commercial partner, or given the financial undertaking that it is, would you like to first find the partner and then proceed? What is your current thinking?
Oh, the answer is the first of all, the financial undertaking. If you look at the average amount of money and time to take a cancer drug to market, we are so frugal and smart in our designs. Inevitably, when a large pharma comes down to speak to me that has interest in the drug, or a big fund manager they inevitably say, to get where we're at, they'd say that would have cost us over $1 billion. You're so good at it into getting to this point in not only oncology applications, but derm applications. We're using a fraction of the money. We run these trials very, very intelligently when we do the designs, what number of patients in there. So the cost is not overwhelming. I recently saw an approved drug in there. I think they used $1.6 billion to get the drug out the door on a very late-stage indication of cancer patients. They're having to charge in excess of $100,000 a dose, a treatment regimen for them, and the poor physicians are having to carry that cost until they can get reimbursed, and so it's dramatically affecting sales, I believe, that it's just too expensive, prohibitively expensive, and so you're not going to have that problem with us. So we can run the trials very, very efficiently, and yes, it would be our intention to start and go forward anyway. And as I've always said, that we intend to hold the oncology as long as we possibly can, and that I would license off the derm applications first. And that would be my first. That strategy has not changed. You never know if someone came in tomorrow and offered us a partnership to purchase it, and the value to my shareholders was correct. Yes, I would proceed forward to license it. But also, from what we were saying, as a co-partner, is not necessarily a trials partner. We can run these trials by ourselves. We have the resources internally offered to us, and we can go forward and run them phase 3s by ourselves if we choose to. But so what we have always thought who our partner was going to be was a marketing partner, that we never intended to build a sales and distribution arm in there, that what our expertise is our novelty of design and clinical trial design and our innovativeness in creating new drugs and how we do this. And so the answer is I can envision us running more phase 2s on different indications, continuing in that route, whereas what we're really looking for in a partner is someone to take over, a marketing partner.
Very good. On that note, Dr. Dees, I wish you the best of luck. The time for our conference call is concluding, and I wish you the best of luck. We will keep in touch as I can write about you on a continuous basis for OMP research. Listeners, do not forget to read the initiation report that OMP Research put out on Provectus Pharmaceuticals in December 2011. Dr. Dees, good luck on your phase 3 trial and good luck on searching for the best partner for Rose Bengal. We'll be in touch later.
Appreciate it. Thank you very much.
Thank you so much.